Hi everyone. So I'm a postdoc in Jack's lab, and one of the overall, I'd say like the main overall goal of Jack's lab, the overarching theme that we all want to happen, is to build a minimal cell, and that's what we call a protocell, that's capable of growth, division, of replication, and eventually selection and evolution. And so most of the people in the lab are actually organic chemists, and they're very talented, and they're trying to get the middle of that diagram there happening. So like genetic material and information replicating without the help of enzymes. But the other aspect is actually packaging this all up, and Marcus gave a very nice introduction earlier, of putting it all inside a compartment so it resembles like life as we know it, cellular life. And so that's where I come in, I work on the membranes. And I'd just like to add that in addition to having a compartment to sort of condense and compartmentalize important things like molecules that might want to react with each other, it's also useful to have a compartment to keep away things like parasites. Say if you're an RNA molecule that's evolved the ability to replicate other RNA molecules, you want to maximize your fitness by only replicating yourself. And so having that membrane there stops parasites from coming in and compromising your fitness. And also having a selectively permeable barrier is nice because you want to be able to, float around in your pond or whatever and take up nutrients, but also not just take up anything. Okay, great. So this picture appeared earlier. So basically, life as we know it is cellular life, and most organisms have cells that are comprised mostly of phospholipids. So these are amphiphilic molecules that consist of a hydrophilic head group, so it has some charges there. That's just a picture. And then you see the two tails that are going out are very fatty chains, and these things actually don't like being in water, and they're fatty acids. And so what happens when you throw this in water is that these molecules spontaneously assemble into a bilayer, and this maximizes the entropy of the system because the water molecules that would otherwise have to awkwardly crowd around the fatty acid tails become liberated to explore space when all the fatty acid tails are actually nestled with each other. And so this is cool, it's a spontaneous process. You can buy like phospholipids from, I know it's kind of expensive, so maybe your lab can buy some phospholipids and then you basically like throw it into water and you see membranous structures form. Okay. Luckily for us, there are much simpler molecules that can also self-assemble into membranes. If you want to make phospholipids in a prebiotically plausible manner, you can give chemists a lot of headaches, I think. But fatty acids are basically soaps. And what happens when we use them as soap is that we're using that at quite a high pH. All of the carboxylic acid head groups are deprotonated, so there's a lot of negative charge. And those charges repel each other, and they want to take up a lot of space, and they form these really sort of high curvature structures that we refer to as micelles. And that's what we use as soap every day. When you buy these from a company, they actually come as a neat oil. So if you drop the pH way down so that all of the carboxylic acid head groups are actually protonated, then all the negative charges are gone, nothing's repelling anything, and they actually sit around as neat oil. And you can pipette it, it's a little bit viscous, but yeah, pretty much an oil. And then the magic happens at this sort of like middle pH, the Goldilocks sort of pH. And you have some head groups that are deprotonated and some that are protonated, and these things can actually hydrogen bond and almost make this sort of two-tailed sort of fake phospholipid-looking structure like I showed you before. But there are no real bonds. There are no covalent bonds. They're just sort of paired up weakly. And you can form bilayers, which is cool. And the reason why we're quite interested in this, other than fatty acids being relatively cheap because we use it as soap, is that they're quite prebiotically plausible routes to synthesizing fatty acids. And if you look at the organic material on carbonaceous chondrites, fatty acids actually comprise, I think, the largest sort of fraction of that. The a lot of the molecules are fatty acids. Okay. Great. So then, like I said earlier, you can throw them into water and they'll just spontaneously assemble into membranes. And what it might look like is what I'm showing on the left here. And we have some structures that are quite dark and visible. And these are multi-lamellar vesicles. That means they have lots and lots of membrane layers kind of stacked up like an onion ring. And they're pretty easy to see under a microscope. You can also get vesicles that just have one sort of cell membrane, or not cell, one membrane, and we refer to these as unilamellar vesicles. Okay. You also get things that are tubular, and then things that are spherical, and all sorts of things. It's quite a zoo. And we can also dye the membranes, red in this case, and then stuff some RNA inside, and the RNA doesn't leak out, the RNA is green. And this is cool. People in lab and other labs have done all sorts of really good experiments using this system. But basically, I'm a physicist by training, so when I look at this, I just think it's too messy. This is what cells look like. They just have that one membrane on the outside, usually quite well separated from any other membranes. And one way of referring to this sort of membrane topology is as giant unilamellar vesicles. So then the giant thing refers to them being so big that you can see them under a microscope. The unilamellar, because there's just that one membrane. And I'm calling this yeast cell vesicle because I work on vesicles, but that's essentially what they are. They're just a fluid sac that's sitting around. That makes beer and stuff. But anyway, pretty useful. So what is kind of crazy is that this is kind of maybe one of the sort of really big challenges in synthetic biology. We want to be able to make these giant unilamellar vesicles to mimic cells, to be able to try out synthetic biology systems, even to use them for drug delivery, so that when they fuse with a cell membrane, you don't have other extra little bits floating around. And in order to be able to make these single unilamellar membranes, people have employed all sorts of different microfluidic techniques. So you need a lot of sort of clean room experience and a lot of, I don't know, it can get quite frustrating, but basically it's a technological challenge to make membranes that are unilamellar. And this is nuts because at some point, I guess it's easy for cells now because cells beget cells, but at some point if you rewind, these unilamellar single membrane forms became favored. And so these primitive cells just did it, like they didn't have microfluidics or anything. And so the thing I wanted to kind of understand was how did this sort of well-separated or unilamellar structure form become favored? And then because I'm a physicist, I'm not going to try and ask, how did this maybe come about because of selective pressures. Instead, I'm going to ask maybe how did this come about from thermodynamics? And then that sort of changes the sort of question you can ask. So instead, I would say what I'm trying to understand is how can we get these things to self-assemble without much, that means no intervention, right? 2 rules, no enzymes. So self-assembly, not do-it-yourself assembly, which is like, you're forcing it into that structure with microfluidics. Okay. So when I say no microfluidics, that's rather specific, and I just mean like we don't want to go into a clean room because that's a lot of work. But it doesn't mean we can't use other tools. And so there aren't really many tools at our disposal in terms of, prebiotic Earth, but we had rocks, and early humans had rocks. And this is a really tenuous line of reasoning, but there was no more logic to it when I tried it in lab. And because I had some rocks sitting around. And I made vesicles as usual, but I put some clay in. And then what you see is that as soon as you add mineral particles, the sorts of vesicles that you form aren't the sort of like high contrast, low contrast sort of variety that you see on the left, but you get these sort of giant rings forming instead. And so that to me was very surprising. And I tried, this is just zoomed in. And then I tried a different way of visualizing this. And this is just, you make some vesicles and then you add a little bit of fluorescent dye to the outside. And the dye isn't able to cross the membrane. So now we're able to see that in the control experiment, when you just dump dump fatty acids into a buffer, they make tiny vesicles. Then a lot of them are multi-layered. We can't really see that structure there, but basically they're not encapsulating much of the liquid. But as you add things like mineral particles, then all of a sudden you're getting much more of the volume being encapsulated. Those are the voids that you see, actually. And so you see this sort of like Swiss cheese-like structure where, there's the same amount of lipid in the image that's on the left, that's on the right. But in the image on the left, a lot of the lipid is wasted in making onion structures, whereas on the right, all of the lipid is being used to make single-layered structures. And so you take up a lot more space. And so this is kind of weird. Like, there's no reason this should have worked, really. There was no logic behind me trying it. But then I think there might actually be a reason. That is that all of these mineral particles sitting around in an organic chemistry lab are there for a reason. They're used for ion exchange or whatever. Basically, cations can like bind to the surfaces of things like morelenite clay, of silicates, and they're usually used to sort of switch out what cation you have or whatever. But maybe what's happening is by adding these mineral particles, the cations are sticking to the clay instead of to my vesicles. And that's allowing the negative charges that are on the surface actually repel each other. And so that's sort of putting the unilamellar structure in, it just makes them more favorable. Okay, so then this is the simple experiment. This is just, vesicles with the usual buffer concentration, 200 millimolar. Almost everyone who works on fatty acid vesicles chooses to use this concentration. It's kind of inherited. But if you just even reduce the salt concentration by 25 millimolar, and I know this here is a different number, but even if you reduce the salt concentration by a little bit, then you start getting these really big vesicles with well-separated membranes. And you can encapsulate dye inside, they don't leak out. Just having one membrane doesn't compromise their sort of integrity. Okay, so I've been claiming that these structures are unilamellar for the last however many minutes. So the final step is just to show that they actually are. And then because making unilamellar vesicles is kind of a big deal in synthetic biology, there's quite a well-developed technique that people use. They throw some dye in. The dye is lipophilic and it goes into the membrane. And the more membranes you have, the brighter they look under a fluorescent microscope. And so on the sample on the right here is made with the high salt buffer conditions. And you can see that in this one field of view, there are multiple vesicles, but they're of different brightnesses. And it's actually quite pretty. And then if you make a low salt concentration sample, then they all look kind of the same, like intensity. So we can write some code to kind of not just eyeball this, but quantify the average intensity of these rings. And you can see for the low salt sample on the left, you get this sort of single peak distribution. And it lines up with like the sort of dimmest rings that we find in the sort of sample on the right that's a bit messier. And so this is kind of pretty good evidence that we've found a way to make these sort of unilamellar structures literally without doing anything. You mix it together, you go to sleep, you come back, and you get the sample on the left. And this is sort of going towards the goal of making these membranous structures that resemble more sort of the life as we know it, like yeast cells. Okay. And I think the reason we're able to do this with fatty acids, and most people in synthetic biology work with phospholipids, is because fatty acids are, have some special properties. The first thing is that they do carry negative charges. Half of the molecules are negatively charged when you can make vesicles, and that really helps the membranes repel each other and like have that separation. And the second reason is that these molecules are small. So they actually move between vesicles, whereas if you make a phospholipid vesicle, the phospholipid is actually kinetically trapped in its vesicle that it happens to be in. It can't move to a neighboring one. And so if you have a complex energy landscape, you can't sort of move anywhere. So basically, these are simple amphiphilic molecules that may have been around on early Earth that can form membranes. They have their advantages. And it can help us form these structures that we couldn't with phospholipids. Also that, having low ionic strength buffers seems to help us form giant unilamellar vesicles, which is cool. And if you can't afford to use low ionic strength buffers, it seems like that you can just chuck some mineral particles in and that'll do the job. Okay, so I'd like to thank everyone here for organizing this. It's a space where, we can all learn a lot. It's low pressure and this is being recorded. Okay, no, we're all very happy. We're all very happy, and we can all be very happy together. No, it's really fun. And I'd like to thank my lab, Jack, Fanny, our lab manager, is amazing, and also to Chris Carr and Kendall Sabota for the olivine that I used. That was actually collected from a green sand beach in Hawaii. Pretty nice. Okay, thank you.
Thank you, Anna. Do we have any questions? Okay.
very fascinating work. I would be curious if is there any temperature dependence of of the phenomena that you observe in your experiment. Also, the other thing for planetary application, I would think that oleic acid is abundant. I don't remember what would be the abundance of oleic acid in meteorite, but probably have you tried some shorter fatty acids like decanoic acid? Would you see some kinds of cell assemblings?
Yeah, I'll answer the second question first because I already forgot the first one. But yeah, so I did use oleic acid everywhere, and its abundance is probably close to nothing on meteorites, right? So this is just like a cheap thing that's easy to work with room temperature in lab. I've tried shorter and shorter chains, and so the sort of behavior where I see these giant unilamellar vesicles form, the pH range at which it happens will change with the fatty acid chain length. So for 14 carbons, I see them form at a lower pH. The problem is when you have these giant vesicles that are about 5 micrometers in diameter. So I'll backtrack a bit. If you pack oranges into a box, the maximum packing fraction you can get is like 60-something%, I think. And so if you calculate the amount of material, of lipid that you need to fill a box, like fill your sample, it is actually only 5 millimolar. And so that's below the critical aggregation concentration for these shorter chain fatty acids, which is why I didn't use them. If you do sort of use more lipid, and instead of them just all forming big vesicles and getting stuck, they start forming like vesicles inside vesicles. And so, I mean, that's a cool behavior in and of itself. But yeah, I think it'd be nice to go to a like shorter chain system like C10 and then just see if we can make well-separated membranes. I'm sorry, I forgot the first part.
One.
Hi, nice talk. Have you ever tried unbuffered system? Like Bayesian is not very prebiotic, and the pKa. What it seems to happen that the giant vesicles are formed around the pKa of the fatty acid. So
Yeah.
if you use the fatty acid as a buffer, have you tried this system?
No, I haven't. The thing that I tried that was the most sort of, the simplest system I tried was no buffer, just hydrochloric acid and sodium chloride, and the same thing happens. It's just annoying to like titrate. So I just stuck to I used buffer instead. Yeah.
Thanks for the great talk. I was just wondering if there was any significant difference in stability of the vesicles between the different concentrations or the addition of minerals.
Yeah, so I didn't actually check their permeabilities to like oligonucleotides or anything. Oh, you mean oh, they're very stable. Like they sit around for like months, and I'm pretty sure the fatty acid I use like has oxidized, and they still look the same under the microscope, and I don't know why. Yeah.
Okay, one more.
I'll echo everyone else. Great talk. So early on, you showed where you guys put RNA inside one of these double membrane things. Could you do the same? Well, first of all, was there any kind of activity with that or purpose, or is it just to kind of show that you could put them in there? And then could you also do the same with the GUVs?
Yeah, so I've tried encapsulation experiments with these GUVs, with the low salt concentration and everything. And the largest thing I've stuffed inside are 400 nanometer large polystyrene particles. And so there's no problem getting nanoparticles inside or like colloidal particles inside, as long as they're diffusing, they'll go in. So there's like huge defects when these things are forming, right? So I don't really know what the encapsulation mechanism is, but you can stuff RNA inside. What people normally do in lab when they put RNA inside is do primer extension experiments. So they're trying to see, can we elongate a primer non-enzymatically? And then they'll usually crack these vesicles open and then run the results in a gel to see if it worked. Yeah, so I think as we move towards developing like optical sort of feedback for successful primer extension, the GUVs will be more useful for that purpose, because then we can like look at something big under a microscope light up. That'll be pretty cool.
Okay, thank you very much, Anna.
Thank you.